folks, let's get started. That is the last time you will hear me say that. Oh, yeah, I know. If you really want to hear it, you can come to my office and I'll say it for you. You know? Alarm. You want an alarm clock to say that? Oh, boy, that would shock people out of their beds, wouldn't it? Okay, let's get started. So, a couple things. Number one thing being shut up. Number two. Okay, so exam in here Thursday morning, 9:30 p.m. a.m. You don't want to come in at night and take this exam? 9:30 a.m. on Thursday morning. Please do as we've done before. Spread yourselves out. If you can't sit every other one, then sit close, and it'll save us getting everything set up. You will have an hour and 50 minutes to do the exam. I'm guessing the exam is going to be about 150 points, so it'll be about 1.5 times as long as the other exams. And format will be the same. And I think that's pretty straightforward. I have not yet set a review session, partly because I'm having some trouble getting the schedule and / location that I want. So what I will do is I will send an email to the class, and I will also post on the web page the time and date of the review session. It will probably be Tuesday or Wednesday evening. I have not decided yet, but that's tentative, so don't hold me to that. I'll let you know when I get something firmed up. It will almost certainly not be this weekend, so it's possible it could be Monday evening also. That's a possibility. Okay, so that's that. I had a question at the beginning is one of the 3 surprises today the fact that I'm not going to talk about immunology? And the answer is no, that's not one of the 3 surprises. So it's nothing that fun. All right. So I've got some distance to go in that, and I want to dig into that. I do have 3 surprises for you today, and one of them isn't too big of a surprise. I have written some songs for you guys, and I've actually written 3 new songs for you. It's the first time I've ever written 3 for one class, which says you guys are cool. You've certainly inspired me to do other things, so I'm very happy to do that. So that's the first surprise, 3 songs today. Okay. Well, this is not a surprise, but it's back to the same old thing. We've got to talk about the immune system. And the immune system is a fascinating, absolutely fascinating aspect of the human body. To put it in perspective, as I mentioned earlier in the term, one of the things that is almost difficult to fathom is the fact that our body can make, in theory, 10 to the 12th different antibodies. 10 to the 12th unique antibodies. And that's in spite of the fact that we only have 10 to the 9th base pairs of DNA, meaning that we have the ability to make 1,000 antibodies for every single base pair in our genome. Now, when people came to grips with that, it was very baffling, because at that time they didn't really understand a lot about how the immune system works and how these different possibilities come about. Today we have a much better handle on that at the molecular level, and I'm going to take you through some of that. But just imagine 1,000 different antibodies for every single nucleotide in your immune system. It's pretty hard to get your head around that. It is certainly for me. When we think about the immune system, we've got really 2 immune systems that I'll be talking about. We have what's called the innate immune system, which is an ancient immune system. It can be found in many organisms, including things like plants. Okay? The innate immune system has proteins that recognize and bind to specific structures, not unlike things that we see in the cell wall of bacteria, for example. Okay? So by latching onto these, that allows the cell to mount a defense against it and then do something about that, perhaps killing the bacterial cell in the process of doing that. Okay? Now, the innate immune system has a series of proteins called toll-like receptors. And toll-like receptors project through the membrane, and they have structures that reach out and grab something. They reach out and grab something. And the grabbing that they do involves something called a PAMP recognition unit. You see a PAMP recognition unit here. There are multiple ones of these. Okay? The PAMP recognition unit has an ability to grab something, and most commonly what it grabs is what's called a lipopolysaccharide. Lipopolysaccharide, as its name implies, involves a lipid attached to a polysaccharide. Bacteria have these things. And so if you can recognize some structures that are unique to bacteria, then you give the cell an advantage of being able to hold on to this bacterium and perhaps do something with it. So that's part of the innate immune system, as I said. And I'm not going to say really much about the innate immune system. That's sort of a schematic structure of a lipid that might be recognized, for example. Well, the more interesting aspect of the immune system is not the innate immune system, but rather the adaptive immune system. And it's the adaptive immune system where we're making antibodies. And antibodies are, as I said, absolutely remarkable in terms of their diversity. So the adaptive immune system uses two types of cells called B cells and T cells. And I'm sure you've heard of these. But before I talk about the cells, I want to talk about the antibodies themselves. Antibodies, I'm sure you've learned in basic biology classes previously, have a sort of a Y-shaped structure. And the Y-shaped structure that antibodies have arises from the combination of what are called heavy chains and light chains. So these are polypeptides that are here. They have a carboxyl terminus. They have an amine terminus. Carboxyl terminus and amine terminus. And so typically what these guys have are two identical heavy chains and two identical light chains. Now, both the heavy chains and the light chains have regions of them that are called constant and variable. So the constant region is found down in the sort of stem of the Y. So if we go from about right here, this is constant, meaning that for a given type of antibody, the amino acid sequence is pretty much the same. The constant region of an antibody is not involved in binding to an antigen. An antigen is, of course, the structure that an antibody binds to. So the antigen binding portion of an antibody is in what's called the variable region of the antibody. The variable region is found out here at these N termini. So both the heavy chains and the light chains have a variable region shown here, also shown on this side. And they have constant regions shown by the C's. So the constant region of the light chain is shown here. The constant region of the heavy chain is shown right there. Make sense? Okay. So that's the general structure of an antibody. And another thing I should point out is these things are held together rather tenuously by disulfide bonds. So disulfide bonds help to stabilize things. They stabilize, as you can see, the heavy light chain interaction. And they also stabilize the heavy chains together. The Y-shaped structure, as this figure tries to show you, is very real. And when you look at this structure, what you see is there's a lot of clustering and clumping down in this region, a lot of clustering and clumping up in these regions, and there's little just joining regions, little tiny regions that help connect these that are fairly exposed. And these fairly exposed regions can be the targets of cleavage by a protease called papain. And if you take and you cleave an antibody that looks like this with papain, what you do is you break it in that sort of joining region, and you discover that, not surprisingly based on what I've told you, this region down here doesn't bind the antigen, but these guys up here will, in fact, bind the antigen. So that tells us, of course, which portion is responsible for antigen binding and recognition. Now, in the immune system is able to adapt. It is able to recognize something as foreign. And I said something. We often think about the something as a protein, but it doesn't have to be a protein. It can be a structure. It could be an invading double-stranded RNA, for example. It could be a polysaccharide that it recognizes as foreign. So there's various things that it can recognize as foreign. If that polysaccharide, for example, is on a transplanted organ and the body says, that's not my polysaccharide, the immune system is going to attack it. And that's where we get the rejection of transplanted organs, for example. So that recognition and that ability to adapt and do that recognition is what I'm going to be spending a decent amount of time here today talking about. Okay. When we look at the different classes of antibodies that are found in our immune system, we discover they can be put into 5 groups. IgM that you see here, which has what's called a pentameric structure, it just simply means there's 5 identical copies of it shown there. IgM is the first antibody that appears during an immune response. Okay? The first one that appears. The first one that appears is not the most abundant one. The most abundant one that appears is known as IgG. Okay? IgA is found in things like tears, secretions, mucus. That's where we find IgA, in those regions there. And you see that they have slightly different appearances as they look there and slightly different connections between each other. IgD, the function isn't completely known. And IgE, if you have asthma or allergic reactions, is one that's very involved in those sorts of phenomena. So we see some specialization of the individual antibodies in terms of location and also in terms of function. I really won't say much about function. But what you notice is that they have different chains that are associated with them. So if I look and I see something has a gamma chain, I say that's an IgG. If I see something that has an alpha chain, I've got an IgA, et cetera, et cetera. And no, you don't have to memorize which one's got which stuff. Okay, questions on that? I'm moving fairly quickly because I've got to get through quite a bit of material. Happy days, right? Okay. Now, if we look at diversity of the amino acid sequence we find in the polypeptide sections of antibodies, we see they are localized to some extent. So we see a lot of variability in the red. We see little variability in the blue. And that's not something we have to be too concerned about, other than the fact that we recognize there is a lot of variable. But this now shows a little bit closer view of that variability that we see. Okay? So again, the regions out here on the ends are identical in a given antibody, and they recognize and bind to the antigen. Now, the binding to the antigen allows the antibody, that is the cell, the B cell that contains it. So these antibodies are attached to cells called B cells. These are projecting out of the B cell cell. And if this guy binds to an antigen, that means, first of all, that it's got the right sequence to bind to that antigen. And a series of events happens that stimulates that B cell to divide and make identical copies of itself. So when a B cell recognizes an antigen, it says, oh, I've got one, I've got it. It's kind of like being the kid in class. I know it, teacher, I know it, I know it, I know it. And the teacher won't look at you, right? Okay? You guys never had that happen, did you? No. The B cell recognizes the antigen, and it stimulates it. There's a process that I'll describe in a minute that stimulates the B cell to divide. And since B cells have unique antibodies on them, it's recognized a specific antibody structure. And when it starts dividing, it's going to be able then to make millions of copies of itself that are all going to target that same structure, because it's going to be carrying that unique sequence that's there. Well, how does it have a unique sequence different from everything else? That's a little hard to understand. Okay? Well, that's what I'm going to talk about at this point. When we look at the antibodies and we look at their sequence, this is a section known as the kappa chains. The kappa chains are involved in some light chain, making a coding for light chain polypeptides. And we're looking right now at the structure of the DNA that codes for this kappa light chain. So we're looking at DNA. And what we discover is that there are various regions called variable regions joining regions and a constant region. In this case, we have a single constant region, meaning that all these light kappa chains are all going to have the same sequence in them. Okay? Now, as we think about this, we say, well, there's these various variable regions. Maybe what the cell is doing in putting this together is it's taking these variable regions and it's mixing and it's matching them. And by mixing and matching them, you get different sequences at the end, such that that gives the cell the ability to make a lot of different antibodies that recognize a lot of different structures. And it turns out that is the case. So you see in this case 40 different possible variable regions that are out there. We see 5 different joining regions. The joining regions are simply joining the variable to the constant. We've got a single constant region. So we can imagine that mixing and matching this with a single one of these is going to give us a lot of possible different antibody sequences. That's one way in which diversity happens, and that diversity is happening as a result of splicing. Remember how splicing, you make a messenger RNA, and you cut pieces out, and you leave pieces in, and now you have different possible ways that you can put those together. So for a given antibody, it's splicing its messenger RNA in one specific way. Okay? If that antibody becomes stimulated to divide, it's going to make new antibodies, all of which splice that RNA in exactly the same way. So all the antibodies that are produced as a result that are going to recognize the same structure and provide immunity, as it were, against that. One of the ways we get diversity is by splicing. A second way we get diversity is unique to cells of the immune system. Okay? In the immune system, immune cells have the ability to recombine within their DNA. Now, recombination I went through fairly briefly in class, but in recombination, what we're seeing is the ability of DNAs to rearrange themselves. So in the immune system, DNAs in those immune cells can rearrange themselves, and you might think of that rearrangement, well, that's a pretty constant thing. Once I rearrange myself, in this case, cut out a section of myself, the section is disappearing from here to here. After I've rejoined it, now I've got fewer things I have to consider for having to splice. So a second way that we make diversity in antibodies is by recombination. Recombination helps in addition to do that mixing and matching, and gives possibilities that might not otherwise be possible for splicing. Those guys together give you about 10 to the 8th to 10 to the 9th different antibodies. But I said there's about 10 to the 12th different antibodies that are possible. How did the remaining ones come about? This one will surprise you. Immune cells can replicate DNA sequences and make mistakes. They have ways of making those mistakes such that that gives another level of diversity. It increases the diversity by a factor of about 1,000. Making mistakes actually works. Once you've got it locked in the DNA, then bang, when you start reproducing that antibody, you're going to make the same one over and over and over. But in generating that antibody, you might make mistakes and get something new. Okay, so that means that your immune system, as the antibodies are being made, have to be doing a lot of mixing and matching of DNA sequences, a lot of miscopying of DNA sequences, a lot of different splicing, such that it makes a finished antibody that now has a unique structure, and that unique structure recognizes and binds to a specific antigen. Everybody with me? This is the quietest day so far. What is this? Are you ready for the second surprise? Okay. Well, the second surprise does not involve a joke, but it does involve something I've never done with a class before. Okay. So the first class I've written 3 songs for, and you're the first class that one of the songs has nothing to do with biochemistry. Nothing to do with biochemistry. Okay. That's good, actually, right? You can actually think, well, we're not going to have to worry about this one for biochemistry. So you've got one of the songs has nothing to do with biochemistry. Okay? I have some fans that are going to come and watch today. Come join us if you would. You'd like to sit down.
You don't want me to see.
This is Dr. Gamble. Dr. Gamble used to teach this class. And he's another surprise. Thank you for coming, Will. Okay, so let's see. Let's get back into finishing up stuff on antibodies here. So, splicing, we've talked about before. You see how the mixing and matching happens during splicing, such that you have different segments that can be joined together. The different segments come together. And finally, you make a finished messenger RNA that now has a unique sequence that will make a unique antibody and will recognize a unique antigen. Heavy chains, not surprisingly, are quite a bit more complex than the light chains. But the same principles hold that we saw before with the light chains. Here's 51 possible variable sequences. Here are another region that we don't see in the light chains called the D region, and we see 27 of those. We see 6 joining regions, and out here we see a bunch of different constant regions. Why do we see different constant regions? That's not exactly constant, is it? The reason we see these, we have different classes of antibodies, and the different classes of antibodies differ in their heavy chain constant regions. So we see different things, different constant regions that can be joined to these variable regions up here to give rise to the different classes of antibodies that I talked about. The IgM, the IgG, the IgA, the IgD, the IgE. Okay? And again, we see the same sort of thing going on. We see mixing and matching. We see recombination. We see error-prone replication. We see all these things coming together to give us a diversity of antibodies that is remarkable. Here's what an antibody sticking off of a B cell looks like. Okay? The antibody sticking outside of a B cell, this is an IgM here. We see that it is in contact, in connection with some other things that are out here. Okay, so these are other membrane proteins that are sticking out and connecting with that antibody structure that's sticking out there. How does the cell know when it binds to an antigen? Well, it's thanks to these interactions that the cell knows. So I'm a cell, I've got this antibody sitting out here. An antibody cell might have 10,000 of these sticking out of its surface. Okay? 10,000. A lot of different antibodies sticking off of that guy. All right? One of them binds, or 2 of them, as in this case, bind an antigen, and what happens? Well, as you can imagine, when we have interaction with a substrate, as we do in an enzyme, we see a slight change in molecular structure of the protein. We see a slight change here. The slight change in structure here is communicated to these guys, which in turn interact with other membrane proteins that in turn signal a process that results in the stimulation of that B cell to divide. Okay? Makes sense? So, once this guy has bound an antigen, he knows it's time to go ahead, go forth, and multiply. Boy, you guys are really, really quiet today. All right. It's going to take something special to get you to wake up. Okay. Here is the different things that can happen. One of the things that happens in the heavy chains is that the constant regions themselves will, in fact, flip out. They will recombine. And when they recombine, we saw a similar thing in the light chains. The recombination results in generating a circle of the removed sequence and then a rejoined, recombined DNA that has an alteration in its combined sequences. Now, this is important, it turns out, because cells need to be able to do what's called class switching. Probably something you think about before you took this class, or maybe the first week or so of this class. You think, if I could do some class switching, I wouldn't be listening to Ahern, right? Maybe not. I don't know. What's going on? Is everybody that worn out for finals? You're that tired? You don't want to listen to the immune system? But we must listen to the immune system. It's an essential part of biochemistry. Yes, Jenny?
So I don't understand why this
question is, it doesn't understand why the recombination gives even more diversity, because the rules for splicing are specific, and it has to happen at a junction, intron-exon junction, that's specific. Recombination is not bound by those rules, and so you could imagine that you might recombine in the middle of exons, such that you create new exons. And so the actual recombination does result in the production of more diversity because of that imprecision of the recombination. So it's like the error-prone recombination, error-prone replication. What's happening is the errors actually give you more diversity. Yes, Megan?
I was just going to ask, so if we're exposed to something for a long time, we can build up antibodies?
If you're exposed to something for a long time, can you build up antibodies? Generally, yes. There are some things, however, that are more immunogenic than others are. So, for example, malaria is something that you can be exposed to for a good deal of your lifetime, and you won't have immunity to it. So it's not very immunogenic. The organism itself also has a lot of variability in the way that it replicates and the proteins that it makes. So those two cause problems. But in general, yes, you're correct. The more exposure you have, if something is immunogenic, yeah, you're probably going to make antibodies against it.
So is that just recombination and the same vectors and things that we're seeing there?
So what you see in immunity, okay, is that you have a group of cells that have been stimulated to divide, and the immune system has a memory. It carries those around and doesn't just get rid of those real quickly. So for many types of immunization that you can have, immunization is thought to give protection, or the immune system has a memory that might last as long as 10 years. So that if I inject you with something that's an antigen for tetanus, for example, then for 10 years your antibody is going to carry around cells capable, I'm doing that a lot, aren't I? Capable of reproducing when they next time encounter that antigen. Okay? Yes, sir?
Could autoimmune disorders come from recombination?
Yeah, autoimmune disorders. Autoimmune disorders happen because of, actually I'll take a minute and talk about this because this is kind of cool and it's a question that students ask a lot. One of the things that happens during your early development of your immune system, when you're a fetus, an infant, for example, before your immune system is fully developed, your immune system is making antibodies against all possible kinds of things, including your own proteins. That's what autoimmune disorders are about, making antibodies against your own structures in your cell. Remember, we had these little license plates or ID tags on our organs that said, hey, this is me. During the development of the immune system, those antibodies that recognize your own structures get destroyed. Okay? So if everything works properly, then you will not go back and remake antibodies against your own structures. When you get an autoimmune disease, what's happening is the antibodies, the immune system is for some reason recognizing part of you as foreign and attacking it. And so that's what's happening with that. Make sense?
Yeah, but could it be just like a random recombination?
It could be. It could be. So again, diversity, there's no absolute guarantee you're not going to make antibodies against yourself. So autoimmune disorders are a concern because you could conceivably be doing that. Yes, sir?
So how would, say, like the defensive venom of the brown recluse cause the body's immune system to attack itself?
His question is, how could the venom of a brown recluse cause the body's immune system to attack itself? And I don't have an answer for that. I don't know. Yes, Jenny?
So does one B cell make all the same kind of antibody, or does it have a whole bunch of different?
So they'll make different ones. Different classes of antibodies. Lord?
How does your body make new antibodies if it senses an antigen that it's never seen before?
Well, okay, so let me answer that question. How does the body recognize or make something against something it hasn't seen before? The body is randomly making antibodies. Randomly making these structures. So during the development of those cells, all these possible things that I said could happen are happening, and your body is throwing out millions of antibodies that it probably will never bind to anything. Okay? Probably will never bind to anything. It's like going and you want to buy lottery tickets. You go out and you buy 5,000 lottery tickets figuring, well, maybe I can get one of these 5,000 that's going to get something. Right? So if I get $1 million and I only invest $5,000, that's, I won't say this is the way I recommend investing, but that's what the body is doing in playing a lottery with the with the antigens. So it's throwing out all these antibodies, If nothing happens, they don't bind anything, they last a few days and they go away. Then another batch of them goes out, etc. But one of them that binds something and recognizes something now reproduces and gets retained in the longer term. Yeah?
So some people tend to get sick more often than other people.
Some people tend to get sick more than others, yes?
Is a possible cause of the people who don't get sick as often that they're producing a greater diversity of antibodies?
It's hard to predict something like that. I mean, you've got diet, you've got immune system, you've got exposure, you've got age. I mean, there's a lot of different things that's there. So I wouldn't ascribe it to any single thing like that. No. Yes, Kyle.
Does anything help to solicit these B cells to come in?
Anything help us?
We have them flowing through, and they're concentrated. They're on very peripheral.
Yeah.
Just what we were talking about last year.
Yeah. No, they're there. And it's all a random process. It's like the process we talked about with, well, how does this thing get over to here? And in the cell, we're talking about at the molecular level diffusion. We're talking about basically random movements that are doing this and random connections that are being made in this sense.
Into our tissues as well.
I'm sorry?
Not just into our system, but our circulation.
Any place where you have any bodily fluid flowing, which is good.
Well,
Yeah. Okay, so let's see. I'm running out of time here. Other questions on that? Vicky?
Why can't like a calf who gets colostrum from mom,
With the what?
a calf that gets colostrum from mom have a better immune system than one that doesn't?
Will a calf that has colostrum from mom have a better immune system than one that doesn't? I believe, and I'm not an expert in the immune system, but I believe that is the case. Certainly if you look at breastfed humans, they tend to be healthier and have a more robust immune system. Also, there's protection during that time that they're getting the breast milk for that. So yes, the answer I would say to that is yes in both cases. No. This is the development of the immune system itself. Yes?
How would antibodies be passed from mom to calf or from mom to?
In the milk.
But how are they not destroyed in the process?
How are they not destroyed in the process? I can't answer that. I can't tell you. I don't know. Okay, you guys are probably ready for the third surprise. The third surprise is the immune system will not appear on your final exam. Now, you're probably sitting here going, well, Ahern, why didn't you say that at the beginning? And I know darn well if I said it at the beginning, nobody would have paid attention to a single word that I said. So I had your full attention as a result of that. So now we're at a point where we can have some fun. So I'd like everybody to relax. Get your vocal cords in action. I hope I have some supporters out here, but just a minute, I've got to do an introduction for them. Give me just a minute to introduce our singers. I have outside singers, and I have some students in this class who are going to help us as well with this. Okay? So, I assume the answer to this is yes. Okay? You guys remember the end of last term, okay? You guys can come in and look at this if you want. You can see that I'm introducing you. So these are the singers today who will be joining us. And, well, in a minute after you get introduced here. This is the only time I've ever brought them in before without a proper introduction. Okay, so this term we've got a very shocking thing. We have people who are into hydrogen bondage. Okay, these are the kinkiest scientists out there, and there's their leader, Kevin Ahern, and the hydrogen bonds. Please welcome the hydrogen bonds. So hydrogen bonds, if you'll all come up. And we are honored to have a guitarist to help us with one of the songs here. This is Kaveen, who I shamed into coming and playing guitar today. So Kaveen, thank you for helping us with this.
Yep.
I've been told by Kaveen I have to stay on the beat, so he's going to help me to do that, I think. Or help us to do that. You want to set up there? Or?
Yes.
Okay.
Because I'll be too loud, right?
Wherever you want. Okay? Okay. All right, so we have three songs. As I said, I've never had three before, so we've got three new songs. The first one is a melody we've sung in this class before. It's to the tune of When Johnny Comes Marching Home. And I'll let Dr. Karplus take a picture of everybody there. This FBI will come looking for us probably. Okay. And it's all about cholesterol. You guys are all worried about cholesterol, right? Naveen, you want to get us started?
Okay.
Naveen. Some things you can build with acetyl-CoAs. I've already done it. Okay.
You guys are late.
All right, get us started again. You can nod your head when it's okay for me to go. How about that?
No. Just feel it, but don't go too
Feel it.
fast.
Feel it.
We're taking chances.
You can build with acetyl-CoAs are joined together partly thanks to thiolase. They come together 1, 2, 3, 6 carbons known as HMG, and you're on your way to mega cholesterol. The mevalonate made in metabolic schemes gets decarboxylated down to isoprenes. They're linked together willy-nilly to build a PP pyranil in the cell's routines to mega cholesterol. There's a single step like Spartan cells, but that's not all. The squalene rearranges to lanosterol. Before that, there's 90 steps to go before the sterols apropos, which you must recall to mega cholesterol. The regulation of the scheme's complex in ways inhibited by feedback of the reductase and statins mimic, so they say, look of HMG-CoA. So we sing their praise and not make cholesterol. Okay. Thank you. That did help to keep us on once I sort of listened to what he was doing. So, okay. So the next one comes from one of my favorite cartoons. You guys know Yabba Dabba Doo, right? All right. So I think we won't have an accompaniment on this one. So we're ready, set, to. Histones, tiny histones, wrap up eukaryotic DNA. Using lysine side chains, they arrange a chromatin array. With them, DNAs of 7 feet fit inside the nucleus so sweet. When you use the histones, you have to deal with condensation and its ablation inside your chromosomes. Now, those of you who were paying attention earlier will notice I said I had one that had nothing to do with biochemistry. And so the last one is I've been kind of inspired by the weather of late. You guys like this weather? You know, there was a song that was written back in the 1920s and 30s when everything was depression and things were bad. And it was called Happy Days Are Here Again. The skies above are clear. Join us again. Let us sing a song of cheer again. Happy days are here again. One more time. Happy days are here again. The skies above are clear again. Let us sing a song of cheer again. Happy days are here again. Now, crappy days are here again. The sky above's not clear again. And the sun has disappeared again. Crappy days are here again. Rain is falling from the sky. I wish I knew the reason why. Guess I'll have to wait until July for the weather to be dry. I do not mean to harangue, since rain provides yin and yang. Because the flowers, everyone, love moisture followed by the sun. Let's stay happy till the rain is done in Corvallis, Oregon. One more time. Happy days are here again. The sky above's not clear again. And the sun has disappeared again. Happy days are here again. Rain is falling from the sky. I wish I knew the reason why. Guess I'll have to wait until July for the weather to be dry. I do not mean to harangue, since rain provides yin and yang. Because the flowers, everyone, love moisture followed by the sun. Let's stay happy till the rain is done in Corvallis, Oregon. Thank you. Please thank the hydrogen bonds for coming and joining us today. You guys have been a great class. I really loved working with you. So see you on Thursday morning at 9:30. I ran out of breath. Thank you, Kavita. I really appreciate it, dude.
Quick question.
Yep.
With autoimmune hemolytic anemia,
Yes.
is it our immune system attacking the red blood cells and making them less, or?
With hemolytic anemia, that's what's happening. I would assume so, but again, I'm not a physician. I couldn't tell you, but that would be my guess as well.
Note cards.
I'll be back on my note card in just a few minutes. So if you want to meet me back there, I'll get you some. How's it? Okay, thank you, sir.
So I lost my note card.
Okay, I'll be back in my office in a few minutes.
So
Okay,
just later.
in a few minutes. Yeah. Note cards. I'll be back in my office for note cards in a few minutes. Yeah, I'll be back in my office in a few minutes. Thanks, Andy. Good. I'll be back in my office in a few minutes. Okay. Yes.
I just wanted to give this to you. Just a little thank you note.
Oh, thank you. Well, that's very kind of you. Okay, picture. All right. Where are we at here?
So here's a seat right here for you.
Me? I'm here. You guys put horns. I can see this
You
one.
can just stand up. It's okay. You sit here and you stand up.
I stand up.
The professor.
Okay.
All right. Well, here I am.
All right.
All right. One more. Somebody blinked, I'm sure. All right. There we go, guys.